Welcome to the Variant Database. Before we begin, I'd like to give a brief introduction. The Variant Database started as a CPI Center for Personalized Immunology initiative back in 2015 with a focus on immunology. Since then, it has expanded to support other new projects such as CACPI, a Chinese collaborative effort related to CPI, mainly focused on lupus, CCG, Canberra Clinical Genomics, the Australian Genomics Health Alliance Genetic Immunology Flagship, and a few other groups as well. Pinpointing the few phenotypically causal variants among the many variants present in the human genome remains a major challenge. In response, we built the Variant Database to facilitate variant analysis, enabling researchers to browse and prioritize variants that may be influential in disease pathogenesis. We currently have over 1,400 patients with over 50 million variants in total and with over 100 users, and we're expected to grow exponentially over the next few years. As a safeguard, these patients are de-identified with assigned study codes, and our web portal is secured using standard web encryption technologies. For our Australian-based users, the preferred way to log in is to use the login via AAF. We use the Australian Access Federation to enable researchers across many different organizations like the ANU, MONUS, etc. to collaborate and access online resources within a shared and trusted environment using their own institutional credentials. This is convenient for researchers who can use their existing username and password to gain access to our platform. And for our international users, or users not formally part of the Australian Access Federation, the preferred way to log in is to use the basic login based on a generic username and password. So let's start by logging in using my account. And we'll have a look at the menus. The most important feature is the search functionality, which we've separated into two types of searches. One is the variant search, and the other is the coverage search. Under the patients menu option, we can search for patients, cohorts, and sample controls. Under the report menu option, we can generate health reports and genotype a panel of variants for sample validation to ensure we have no sample mix-ups. We also have something called search profiles, enabling users to quickly populate commonly used search filters that are used over and over again, and it's there for convenience. So let's start by having a look at variant search. On the search page, users might be intimidated by the range of options available, but this also makes it a very powerful search tool. So I'll just go over the filters very quickly. We can filter on patient details, like their study code, their affected status, and we can filter on list of genes. We can filter on zygosity, homozygous or heterozygous. We can filter on the types of mutations, for example, exon, splice, missense, nonsense. Frequencies such as exact, nomad frequency. Genomic location based on their chromosome and coordinates. Predictive tools such as polyphen, SIFT, and ClinVar. And the next filter is particularly powerful, enabling researchers to filter for variants that are shared between individuals or variants that are unique or de novo, classified as de novo. For the sharing of variants, it doesn't have to be 100% found across all the individuals. Users can even set a percentage of sharing, which is useful for conditions which are thought to be polygenic. We have filters for pedigree analysis that can pick out variants likely to follow Mendelian inheritance, including compound hets. We have two ways to filter on pedigree analysis. One is pre-analyzed by the pipeline based on known related individuals at the time of analysis. And this makes search very quick. And the other one is real-time pedigree analysis based on the most up-to-date known relatives, which can take up to 20 minutes to complete. We can filter on read frequencies, the var allele frequency and ref allele frequency. We can filter on known variation using db snip rs numbers. We have another database which collects patient information, including phenotypes. If we've collected phenotypic information for the patients of interest, then we can filter variants based on phenotypes. We have a pipeline-specific filters such as bioinformatics ID, which is shown as the cohort or single IDs, filters for structural variation, filters for the reactome pathways, and filters specific to primary immune deficiencies. Now I'll go through a typical example of how these filters might be used. Based on the phenotypes expressed by the patient, we may have a list of predefined genes we are interested in. And if that's the case, we can populate our search with a list of genes by simply copying and pasting from an Excel spreadsheet. So I've got a list here already, and I'll just copy and paste a small list as an example, like so. Users that repeatedly use the same gene list over and over again can create something called a search profile. And I can just simply give it a name, my gene list, and we'll just paste the genes here, save. And we can go back to the search and select our, I'll just expand the genes filters here, and then I'll select from the drop down my gene list and click populate, and you can see that it's populated the gene list here. It's really convenient, especially for users that are investigating multiple diseases with different gene lists. Okay, so let's take a step back and say, well, what if we didn't know what genes to use? And that's okay. We can use the phenotypes of the patients and consult with OMIM to come up with our gene list. And so let's have a look at the phenotypes of our first patient from the 1000 Genomes project. So we can see this patient has phenotypes lipoma, thyroid adenoma, and macrocephaly. The phenotypes were populated in a separate database, which we pull in to provide integrated searches, as we will soon show. So I'm just going to copy and paste this study code, and going back to our search, we can paste in the study code here. Then scrolling down to phenotypes, we can tick the phenotypes gene checkbox and then click search. So this may take around 30 seconds or so, as it queries OMIM in real time for a list of genes to search on and applies the variant search in one step. This is essentially a phenotype to genotype approach. We can see there are 22 variants associated with our patient's phenotype. So we have 3 tabbed results to look at. The first tab is the list of variants. The second tab lists any genes which has poor coverage, either in the sequencing or variant calling process. This way, users can browse variants and the coverage results side by side, providing a broader view over the quality of the variants being presented. Importantly, this would be useful for difficult to diagnose patients for which no causal variants have been identified, where potentially disease-causing variants may lay hidden in uncovered regions of the genome. In such cases where coverage is poor, we may want to consider resequencing with greater coverage. And indeed, we have a few cases where we've seen this happen. And in the third tab, we have the phenotype to genotype results, where we list the phenotypes, genes, and modes of inheritance as reported by OMIM. We can also export our 22 variants to Excel for offline analysis. Also, we can annotate our export with additional information by calculating on the fly the CA-CPIC frequency based on our in-house Chinese controls. Also, we can add nomad frequencies for all ethnicities. And keep in mind that the more options you add, the slower the export, as all of this is done on the fly. Okay, so let's filter our list of 22 variants even further using population frequencies. And we'll set our nomad frequency in particular to less than 1% for rare variants, and click search. And there we have found our causal variant in the PETN gene, which is associated with the confirmed diagnosis of Cowden syndrome. So let's look at this variant in more detail. So at the top of the page, we have our basic patient details, including study code, cohort ID, affected status, and listed phenotypes. We can also link out to our patient database by clicking here to get more information on this patient. We also list known diseases associated with this variant as reported by other public databases, and this includes DB-Snip, ClinVar, and we also list genome-wide association studies, if any are found. Further down, we have details about its sequencing parameters, such as exome or whole genome, read length, the instrument that was used, single or paired, and the date when the sequencing was first requested. Next, we have variant annotations in two separate tabs. By default, we show the annotations from our bioinformatics pipeline, which includes gene, genomic coordinates, ensemble transcripts, protein domains, uniprot, mouse phenotypes, gene ontologies. The more important ones include mutation type, in this case, which is a nonsense mutation. There's also read depth, amino acid change, the calculated CA-CPI control frequencies, read allele frequencies here, the read allele and its associated count, the var allele and its associated count as well. And further down, we have the pipeline prediction and scores, which contains polyphen, SIFT, CAD-FRED, variant score, and the ClinVar reported pathogenicity. While the bioinformatics pipeline does its best to stay fresh, it's optimized for performance using pre-downloaded annotations based on the older GRCH37 assembly. For the more recent annotations, we have a separate tab called Latest Annotations, which will download directly from Ensembl on the fly the more recent annotations. Also, the data presented here is in a slightly different format with some additional annotations. Here we present each individual Ensembl transcript along with GURP scores, mutation taster scores, and sci-fi scores. The next section is sample availability and is primarily used by our ANU-based users where we track all samples associated with the patient and where they're stored in a separate sample storage database. In the next section, we have our downloadable datasets, which include VCFs, BAMs, the TSV reports in Excel format, and summary stats reports reported by the pipeline. In this example, we can see that the BAM file has already been archived to tape. Our current policy is that any BAM file older than a year old is automatically archived by our system. And we do this because BAM files are notoriously large and conserving disk space has been a major challenge. However, if users would like to still have access to their BAM file, it can be restored at a click of a button here. Users will automatically get an email to notify them that the BAM file has been restored and ready for download or IGV viewing. But please note, it can take a few hours to completely restore a BAM file. And checking BAM files is necessary to eliminate the high number of false positives and can be used with the IGV viewer developed by Broad Institute. Next, we show if anybody else may carry the same variant based on the user's assigned access. Access to patient data is controlled by a user's assigned teams. Some patients can be shared across multiple teams for data sharing. And we can see that for this patient, access is restricted to the users belonging to the CCG team only. Down at the bottom, we see a button called Save as Provisional Variant. This is useful for users that wish to compile a list of candidate variants, but not yet confirmed. The variant details are transferred over to the patient database, where the full list can be viewed on a per patient level. Now let's come up with an example of poor exon coverage. Just going back to the search page. And how we can interpret them. So using my predefined list, just double check that's populated, and paste in the study code, and we'll just apply the search. All right, and so we can see that clicking on the exon coverage tab, there's a warning icon. And we can see that there was one gene that had fairly low coverage of only approximately 34 base pairs. If this gene was a candidate, we may want to consider resequencing with greater coverage, perhaps as whole genome. We can also generate a full health report for an individual. From the menu, click on Reports and enter the patient study code. I'll do that here. And we can specify which class of information we want to include in the report, such as ClinVar, genome-wide association studies, and Snippedia. Since the report usually takes around 15 minutes to generate an email, will be sent along with the attached health report. So I'll open up an example. Across the top we have 3 headers. Just scrolling back, we have Snippedia, we have ClinVar, and we have our GWAS studies. If we click on clinical significance and filter down for pathogenic, or pathogenic, likely pathogenic, we can see our variant with the PTEN gene highlighted here. We can also look at GWAS studies. So under the GWAS section, I'll select everything except blanks. And so we can find any statistical association with variants and the patient's phenotype, essentially a genotype to phenotype approach. Just a word of caution, we have to be careful in interpreting these reports, treating these as potential risk factors for disease only. Alright, so the example just shown is for analysis of a single patient. Let's look at a more complex example with a trio family from the 1000 Genome Project, which we have given a cohort ID of CCG cohort 1. So going down to our pipeline filter, and we'll just do a quick search. And we'll just pick any variant at random. We can see now we are presented with the pedigree diagram of the family relationships between individuals. And just below that there is a table showing which individuals carry the variant, who's affected, and predicted zygosity, along with any read frequencies if they're available. Going back to our gene search, let's say we found nothing of interest based on our gene list. So as an example, let's search on our cohort based on 3 genes. So I'll just truncate this gene list down and filter on our cohort. Our search results returned only 12 variants, none of which looks interesting, with mostly pretty high frequencies. So going back to our filter, we can click on gene interactions, and now we can click search again and see what we get. So now we have found 270 variants, some of which look more interesting with lower population frequencies. To view the expanded gene list, we can click on the gene filter, and we can see our original gene list on the left hand side and the expanded gene to gene interaction list on the right hand side. Another thing we can do is filter by phenotype. So we can just click on the Add Phenotype Filter button, and we can apply search. And we'll just use systemic lupus erythematosus as an example. And we can click yes and add. And you can add more phenotypes if you wish. And that'll filter your variant search according to patient phenotype. Next, we'll look at Snip validation under the genotyping menu. So the process of sequencing and analysis goes through a series of steps with quite a bit of human involvement, and therefore is prone to error. In order to verify that a patient indeed does have a particular variant, we can go through a step of Snip validation for a batch of patients. And so we've created a tool that can be invoked through our web portal to determine the smallest combination of SNPs required to uniquely identify a patient within a batch for a predefined SNP panel. And so this process requires that a BAM file be available and can take a while depending on the size of the batch. So let's open up an example. At the top we can see our SNP panel with individuals going down the rows and their expected genotypes going across with the genomic positions. Further down we've compiled a combination of SNPs for each individual to uniquely identify them. And in this example we only need a maximum of 2 SNPs per individual. As batches get larger, determining which minimal set of SNPs to be used can be time consuming and error prone, so we've automated this step for our technicians. And finally, the database is a fast evolving system with frequent updates, and therefore it's worthwhile to check for updates to see if there's any new features which could be useful. From the menu you can hover over the docs and click on release notes, and here we blog about our updates. You can go directly to our blog site, and users can subscribe to receive email updates. Thank you for watching.